Ma'am, the recording is already started, like it is there, recording.
Molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into
Sound is
useful
coming.
energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed
You
from
can
the
hear the
consumed
sound?
food and transported into the
Yes,
bloodstream.
ma'am.
From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
We have this entire misunderstanding of what Darwin really meant when he said survival of the
Just go
fittest.
through this 3-minute video where you can differentiate the aerobic and anaerobic
Every organism needs energy
respiration.
to survive. For human beings, food is the source of energy, and it requires oxygen to release this energy. Respiration is the process by which the body obtains and utilizes oxygen to produce energy inside a human's body. There are 3 steps of respiration in humans. Breathing is the process of moving air into and out of the lungs to facilitate gas exchange with the internal environment. This brings in oxygen from the air and flushes out carbon dioxide. Internal respiration is the exchange of substances between blood and cells, whereas cellular respiration is the release of energy from food substances in living cells. Today, we will discuss cellular respiration only. Cellular respiration. There are 2 kinds of cellular respiration, aerobic and anaerobic. Aerobic respiration is the process by which oxygen-breathing creatures turn fuel, such as fats or sugars, into energy by the help of oxygen. The key organelle inside cells of aerobic respiration is the mitochondria, where the glucose molecule from the food goes through a multi-step process, such as glycolysis, Krebs cycle, and electron transport chain. At the end of this multi-step process, 1 molecule of glucose, with the help of 6 molecules of oxygen, can produce 36 molecules of adenosine triphosphate, or ATP, along with 6 carbon dioxide molecules and 6 water molecules. This makes aerobic respiration a very efficient process. All multicellular organisms and some yeast can carry this type of respiration. Aerobic respiration is needed by many processes in our body to run properly. Muscle contraction, such as in limbs, cardiac, and peristaltic motion, need the ATP generated through respiration as their fuel. Formation of peptide bonds in protein synthesis, the synthesis of chromosome and cell membranes, cell division in growth, transmission of nerve impulse along the axon by transporting sodium ions in and out of the membrane, and regulation of body temperature are all powered by the aerobic respiration. The examples of anaerobic respiration are alcoholic fermentation, lactic acid fermentation, and process rigorous muscular activity. Alcoholic fermentation is carried out in plants and some yeast, Saccharomyces cerevisiae, which sugar is broken down to release energy. 1 molecule of glucose turns into 2 ethanol, 2 carbon dioxide, and energy of 2 ATP is released. This process is an economically important process that is used in bread making and brewing beer and wine. Some bacteria, such as Lactobacillus bulgaricus, cause milk to turn sour and form yogurt. The bacteria feed on sugar, which is then converted into lactic acid and energy. This energy is also equivalent to 2 ATP. The bacteria turn glucose into 2 lactic acid and energy of 2 ATP. In human beings, during strenuous activity, breathing is not able to provide sufficient oxygen for respiration. Muscles experience a shortage of oxygen, so the aerobic process becomes anaerobic. This change causes formation of lactic acid. Accumulation of lactic acid causes muscular cramps and fatigue as a safety measure to protect the muscle cells. Rapid breathing after the activity helps to repay debt by increasing oxygen in the muscles, thus converting the process back to aerobic.
Hold on. There are confusion in exorganic and endorganic reaction, na? Let me show one short one.
So from the very start, we have been on Shopify. We've built the website all by ourselves. Hi, that's Gayatri. And that's Ankita. And our brand is called Dare. I think it's been a very seamless process setting up a shop.
So let's talk about two different types of reactions.
Can you see the video? New one. Two graphs are seen.
Yes, ma'am.
Just check.
That can occur in a cell. The first is the exergonic reaction, and the second is the endergonic reaction. And you can see by the graphs of each of these types that we have, first of all, on the y-axis, we have energy that's increasing, and then along the x-axis, we have just time. So in other words, this is the progression of the reaction along the x-axis, and this way energy is increasing going up. So for a reaction, okay, we have reactants, okay, as our starting material, and we end up with products. So if we look at the exergonic reaction, what we see is we have a certain amount of energy that's potential energy in the bonds of the reactant molecules. And we have a certain amount of energy that's in the bonds of the and provide potential energy in the bonds of the product molecules. And you can see that in an exergonic reaction, there is more energy in the bonds of the reactants than there are in the bonds of the products. But we know that energy can't be created or destroyed, therefore that energy is going somewhere. So in this case, energy is going to be released in the cell. Now the reason that's important in biology is...
Understood this point? Or should I repeat this one? Just repeat this one.
Ma'am, I
Why
think this.
energy is
in the
released?
bonds of the reactants than there are in the bonds of the products. But we know that energy can't be created or destroyed the product molecules. And we have a certain amount of energy that's in the bonds of the and provide potential energy in the bonds of the product molecules. And you can see that in an exergonic reaction, there is more energy in the bonds of the reactants than there are in the bonds of the products. But we know that energy can't be created or destroyed, therefore that energy is going somewhere. So in this case, energy is going to be released in the cell.
So in case of exorganic, the product energy is less and the reactant energy was high. So when high to low, the energy is decreasing, so that energy will be lost somewhere. This losing energy, the lost energy, is visualized as the energy release. Understood?
Now the reason that's important in biology is because that energy can be used for the cell to do work. So this, in other words, an exergonic reaction can be put together with or coupled with another reaction that requires energy to drive that reaction forward. Now if we look at the other type of reaction over here, an endergonic reaction, okay, it's the exact opposite. So if you look at the amount of energy in the reactants, in the potential energy of the reactants versus the products, we see that there's more energy in the bonds of the products. Well, that has to come from somewhere. Therefore, for this reaction to go forward, there's going to have to be an input of energy. So the big difference, an exergonic reaction is going to release energy for the cell, and an endergonic reaction is going to require an input of energy into the cell. So these two types of reactions can be coupled together, so an exergonic reaction can drive an endergonic reaction in the cell. Now, I want to point out something that you should see that's common to both of these reactions, and that is that the reaction doesn't go directly from the amount of energy in the products down to the energy, reactants to the products, okay? And it doesn't go directly from reactants to products here. Instead, we have this sort of hump of energy that has to be overcome in order for the reaction to go forward. Okay, that energy is called the energy of activation or activation energy. And this is something that is important when we talk about enzymes, because enzymes are going to actually lower the amount of activation energy it takes for a reaction to go forward.
Understood.
So let's look at our same energy diagram. So we have time still this way, right, as the reaction progresses
Why
forward.
energy to
And
reduce
again, we have
the
energy
consumption of the
increasing
energy?
on the
Why
y-axis.
enzymes are
And if you'll notice in red,
required?
it shows the amount of activation energy that would be required. Now look, this is an exergonic reaction. So this is a reaction that energy is being released. It does not require, it's not an endergonic, okay? But there's a certain amount of energy that needs to be provided in order for the reaction to go forward, okay, to get it started, if you will. Now notice with an enzyme, if we follow the black line, it takes much less energy or activation energy in order for this reaction to go forward. And so that's why you will read that an enzyme is a catalyst. In other words, it's getting the reaction moving forward. Now, an important thing to know is an enzyme is not used up in the reaction, okay? An enzyme can assist in a reaction and then turn around and do the same thing again. It is not in any way used up like you might think of a catalyst.
It is a helper. Enzyme
For
is a
example,
helper
lighter
which
fluid that
reduces
starts
the
a charcoal
energy
fire.
consumption.
That catalyst is
Or
used up, but an enzyme is not used up.
Understood.
Now let's talk a little more about what an enzyme is and how it works in the cell. Okay, first of all, an enzyme is a type of protein.
Well,
Ma'am, this we all know, it is not
so you
anything.
don't need, okay.
Yes, ma'am. This part actually we have done it for earlier.
Okay. So for this chapter, anything has to be understood more? Do you have any query?
No, ma'am.
Okay. So all the best.
No, ma'am.
When 26th is the exam, prepare well.
Ma'am, also stop the recording.
Okay, okay.
Ma'am, how many questions then?
Dara Baba, Dara.